Population genetics. Scientists building on the research of Darwin and Mendel came up with a whole new branch of biology called population genetics. Population genetics examines how the frequencies of alleles in populations change over time. The inheritance patterns and principles found by Mendel explained how variation occurred in populations. Natural selection explained why some alleles became more frequent in a population than others. A population's gene pool consists of all the genes in all the individuals in that population. Each variation of a gene has a gene frequency in the population. The gene frequency is the ratio of an allele to the total of all other alleles of the same gene in the population. This frequency can be measured and then compared to its frequency in past or future generations. If there is a change in gene frequency of the gene pool, then the population is evolving. Genetic diversity occurs when there's a great variety of genomes within a population of organisms. Populations having genetic diversity can evolve by natural selection. Genetic diversity allows populations to adapt to ever-changing environments, as they are more likely to contain individuals that can survive the change. Surviving individuals will pass on their alleles to the next generation, and the population evolves to meet the new environment. Populations with little or no genetic diversity face extinction ultimately, as the environment changes and the individuals cannot adapt. However, should the environment change, as they often do, the evolved population's alleles might not be helpful and might even be harmful. Once again, genetic diversity will help the population survive. All the examples so far are of evolving populations, as that has been Darwin's hypothesis. But good science requires that when attempting to prove a hypothesis, the results that would occur if the hypothesis is incorrect must be determined. This provides a standard to compare with the experimental results. The opposite hypothesis is called the null hypothesis. For evolving populations, our hypotheses are H1, populations evolve by descent with modifications as proposed by Darwin. H0, populations do not evolve by descent with modification as proposed by Darwin. The null hypothesis must be quantifiable. For example, if proposing that a coin has been tampered with, causing it to be more likely to be heads when flipped, the null hypothesis is that there will be exactly 50% heads and 50% tails. This is more complicated with topics like evolution, so a specific measurable trait must be focused on and quantifiable tests must be applied. The Hardy-Weinberg equation and the chi-squared test will be used in evaluating if a population is evolving. We'll describe the Hardy-Weinberg equation later in this presentation. The chi-squared test was described in the scientific methods unit. Consider the following event. In 1932, a large pine tree forest in upstate New York suddenly began to die. Scientists identified a disease microbe that was responsible but could not find a cure. In 5 years, 96% of the original population perished. A scientist noticed that some of the trees were unaffected and began to record data. He observed that the surviving trees began to produce new trees quickly. Within 50 years, the pine tree forest returned to its original population size. Then, in 2005, a few trees perished in the same forest. Fearing the worst, biologists tested the dead trees and found the disease had returned. However, by 2010, only about 10% of the trees perished. Scientists hypothesized that the 1932 survivors had a genetic mutation that produced an allele that shielded the trees from the infection. Since the 2005 forests were all descendants of the survivors, most carried the allele. The hypothesis was verified by genetically sequencing a tree that died from the disease and comparing the alleles to an immune tree. The allele that protected the trees was identified. The researchers labeled the alleles D positive or D plus for having the mutation that causes immunity and D negative for no immunity. Controlled test crosses were performed to understand why some of the trees died, even though they were all descendants of the immune trees. The researchers theorized that the immunity allele was dominant to the old, and the old allele was still present in heterozygotes of the population. So we have this heterozygous D positive and D negative in the population. These individuals would be immune, but possibly produce offspring that were not. First, to check out the null hypothesis, 2 non-immune trees were crossed to see if they would produce immune descendants. The Punnett square indicates that they would not produce immune descendants, and this was verified by experiment. So you can see crossing 2 non-immune plants produces seeds that are also not immune. The researchers tested the hypothesis by identifying heterozygotes by random crosses of the immune trees. When both parents were heterozygous, some non-immune trees would be produced. The Punnett square predicts 1 out of 4 non-immune trees. So if you cross 1 heterozygote, dominant, with 1 homozygous dominant, you get all immune seeds, okay? All daughter trees have immunity. Crossing 2 heterozygous trees, this tree would not have the immunity even though both its parents had immunity.